Good evening, one and all gather here. I welcome you all to this 22nd session 2 CSIR Life Science question and answer discussion session. Morning we are discussed about the Unit 1 questions part C. Today this session is full on the Unit 2 cellular organization. Till now we have discussed 4 units, that is Unit 4, 5, 13 and first unit. Hereafter we are having 8 more days and we are having nearly the 8 subjects. So the 8 units. So only I am planning this evening session. Due to some my personal works, I was missed the past 1 week session. I will cover the unit in this week itself. Today only we are having evening session. Tomorrow onwards I will ready for morning session. Please be with ready with on time. Okay. Today this is second unit, that is cellular organization. In this cellular organization, major topic is the it's a major topic in the part B session. Okay. As I mentioned in the earlier classes, unit 4, 5, 13. If you are finishing this 3 units, 4, 5 and 13, you can achieve nearly 100 marks. And this unit 1, 2, 6, 7, these units are helpful to achieve the 100 marks. So 4 units are there. And these are all the biggest units, and it's all very important in the 2 mark B sessions. And part C questions also, these are all very important. In another hand, it's another way. If you are studying for a part C, you can easily clear the part B also. Okay. Here the first part, this is all the part C questions of the unit 2. The budding yeast cells that are deficient for MAD2. It's a deficient. It's a protein compound deficient for this particular protein compound. This compound, a component of spindle attachment check protein, are killed by treatment with the phenomenon which causes microtubule to depolymerase. Okay, here the statement they are given. The budding yeast cells that are deficient of particular compound, that is MAD2. That compound of the this compound is a spindle attachment checkpoint, are killed by treatment with the phenomenon. This can cause microtubule to depolymerase. In the absence of phenomenon, okay, there is the absence of phenomenon. However, the cells are preferably viable, that are in a viable condition. Which explanation out of the following is able to justify the observation? Here they mention the concept. They provided the question. What they mentioned, there was some pro-yeast that is having some lacking of compound. If that compound is, it's a very, very important checkpoint. This checkpoint they are killed by the treatment of phenomyl. So the cells are dying because of treatment of phenomyl. This can helps to depolymerize. This is the statement. As per their statement, what they asked: if there is absence of phenomyl, this, however, cells were probably viable. Okay, they are in a live condition. Which explanation out of the following is able to justify this observation? In the absence, the answer is A. In the absence of phenomena, the majority of spindle form normally, and the spindle attachment checkpoint MAD play no role, because that is the confusion statement here, that play no role in this cell viability. So that the main point is the majority of the spindle from the spindle form they are normally formed. There is no role of the MAD2. And next question, during receptor mediator endocytosis. This endocytosis process, there are no in the cell system, there is called receptor mediator endocytosis is very important. In this receptor mediator endocytosis, the apolipoprotein B. This in this apolipoprotein, they are present on the surface of LDL particle and binds on the LDL receptor present in coated pit containing clathrin. In this concept, they mention during this receptor mediator endocytosis. It's this major thing here is in this receptor mediator endocytosis is a major present component is clathrin. Okay, this clathrin is this endocytosis is also called as this clathrin mediator endocytosis process. In this process, the cell surface protein, okay, that captures the specific target molecule and then pass the signal to transmembrane proteins and makes the the cluster from the plasma membranes are attached to the coated protein. Okay. This concept that signals is passed to inside because of the clathrin molecule. Here they added the apolipoprotein B. These are apolipoprotein B that on the surface of LDL particle binds to the LDL receptor present in the coated pit containing clathrin. The receptor LDL complex is internalized by endocytosis, trafficked to lysosome, and the LDL receptor is finally recycled. These are all the sequential process. First one is the receptor LDL complex is internalized by the endocytosis and then trafficked by lysosome, and LDL receptor is finally recycled. This is the step. And the patient report with normal hypercholesterolemia. Okay, here what they ask, this could be due to why, what is the reason? Means the here they mention the mutation is the mutation in the LDL receptor. So only the patients are shown with the hypercholesterolemia. Here this LDL receptor is finally recycled. But after this recycling process, that signaling is not recognized, the lipomolecule. That is, there is a mutation on the receptor. So only that not getting the signal of apolipoprotein. So lagging of apolipoprotein, there was hypercholesterolemia happened in our body. It's very easiest and direct question. If the receptor recognizes the apolipoprotein, our cholesterol system will get degraded and there is no normal cholesterol level shown in a patient. If there is a mutation in the LDL receptor, there is no signals in the receptor. So there is no possibility of apolipoprotein binding. This lagging of apolipoprotein binding, it makes a over cholesterol in the blood. So they are called, their report is shown hypercholesterolemia report. Okay, a logical question here. Overall length of cell cycle can measure from the doubling time of population of exponentially profiling cell. Here the mention here is the length of cell cycle can be measured from the doubling time of population. Doubling type of population of exponentially profiling cells. Exponential means that is in the exponential phase. That cell growth having lag, log, and exponential and death phase, right? That cells are in the exponential phase. That is the growing phase. In this, what they asked, what is the overall length of cell cycle in a mouse L cells? they mention the doubling time of population of mouse L cells are determined by counting the number of cells in the sample of culture. Okay, if they're counting the sampling culture, and likewise they are found the cell cycle length. Okay, if the cell cycle increases, means cell count become increase. That is concept here. Cell cycles are increases means you can get more cells. In the first cycle, if you have 10 cells, means in the first cycle that will get double. After that you will get 20 cells. Next step, 20 cells are going to divide and get 40 cells. These are the concepts. And the next concept here is they are asked what is the overall length of the cell cycle in mouse L cells. In the mouse L cells, okay, what they asked here, they mentioned the timing 20 hours, 40 hours, 60 hours, 80 hours, and 100 hours. And the number of cells is initially they are having 10 power 5, and it's doubling. It gets double in the 10 power 7 and 10 power 6. It will increase in the time permit. Initially, it is very less number. During that the process of time, that will increase. So they are asked what are overall length of cell cycle. As per their question, the overall length of cell cycle should be a 10 hour. It's initiated from here. That in this 10 hour, that after the cell cycle, that will divide. Each and every 10 hour that will get divided. Okay, during receptor, mediator, and this is the same question as such what I included in the previous. So go to 38th question. Here the eukaryotic compound, eukaryotic chromatin, 30 nanometer fiber can open up to give rise to two kinds of chromatin. There are two chromatins are there. In this two chromatins, in one type A, the promoter of gene with open chromatin is occupied by the nucleosome. So the A, okay, one type 2. There are two types kind of chromatins are there. The A type promotes a gene within open chromatin is occupied by the nucleosome. The next in the other B, okay, that present in the B, the promoter is occupied by histone H1. Okay, this is a statement. It's given in the cellular arrangement. The main thing whenever you are starting the cell cycle, cell process related study, the genome architecture, you want to study the genome architecture. It is well explained in the genetic engineering book Primrose. Okay, this is a very important thing in the this genetic engineering and cellular signaling processes. And one more thing here is the eukaryotic chromatin that have in their mention, that is the fiber solenoid, can open up to give rise to two kind of chromatins. In one type A that promotes a gene within an open chromatin is occupied by nucleosome, whereas in the other B. So that is packed with, that is occupied with histone H1. Here what they asked, which of the following set is correct? Gene A, that is the gene in A is repressed. Another one is the gene in a B is activated. So in this case, what is the possibility? What is the output? What is the expected result? Means the gene in A is repressed, gene in B is activates in active mode. Okay. In this erythrocyte membrane cytoskeleton consists of mesh work of protein underlying the membrane. This is the Just a little bit advanced question. Erythrocyte membrane skeletal consists of mesh of a protein overlaying the membrane. The principal compound spectrin has alpha and beta subunits. Okay, here they mention the principal compound alpha has that having the two subunits, that is alpha beta subunits, which assemble to form a tetramer. Then cytoskeleton is anchored to the membrane through linkage with transmembrane protein bond 3 and glycoprotein C. These two things that happen. And cytoskeletal domain of band 3 also serve as a binding site of glycolytic enzymes such as glyceraldehyde 3-phosphate dehydrogenase. This is the enzyme which they used. Glyceraldehyde 3-phosphate dehydrogenase. Analysis of blood sample of a patient with a hemolytic anemia show spherical rod red cells. The patient carries what is the answer means, that is a C, mutant beta spectrin infective in alpha beta dimerization ability. Okay, here they mentioned the mutation happened. Here read the story. As I mentioned in the previous classes, the CSIR having a biggest stories and easiest answer. The answer present in the question itself. The erythrocyte membrane skeleton consists of network of protein underlying the membrane. This is just the intro, not necessary for this question. The principal component spectrin. Okay, this is the compound. Spectrin has a two subunits, alpha and beta. It's assembled to form a tetramer. The cytoskeleton is anchored to the membrane through the linkage between the transmembrane protein. There are two membrane proteins are there, band 3 and glycoprotein. This cytosolic domain of band 3 also serves as a binding site of glycolytic enzyme that such as glyceraldehyde 3-phosphate dehydrogenase. So what is there asked? Analysis of blood sample of patients with a hemolytic anemia. Hemolytic anemia. In this hemolytic anemia, the cells are degraded. But in that sample, the blood samples are shown the spherical. They can see the spherical blood cells. How they can see? If there is mutation in beta spectrin defective, that if there is a mutation in the beta spectrin or spectrin that don't have ability to dimerize, so alpha beta dimerization ability, it's defected. Okay, these are all the concept. And next question is maturation promotion factor MPF control the initiation of mitosis in eukaryotic cells. This is the just hint. MPF kinase activity requires cyclin B. If you want to do this process, that is the maturation promotion process, we need MPF kinase. That MPF kinase needs cyclin B. Cyclin B requires chromosome condensation, and breakdown of the nuclear envelope into vesicles. This cyclin B degradation is followed by chromosome condensation, decondensation, nuclear envelope reformation, and exit from mitosis. This requires ubiquitous of cyclin destruction box motif in the cyclin B. This is the statement here, the keyword here. An RNase treated Xenophus egg extract and sperm chromatin were mixed. MPF activity increases with the chromosome condensation and nuclear envelope breakdown, where, however, this was not followed by chromosome decondensation and nuclear envelope reformation. Because they asked the answer D is cyclin B lacking the cyclin destruction box has been overexpressed. They tell the story and ask the question from the story. That itself in this both questions are the story based question. So first step is the Maturation promotion factor controls the initiation of mitosis in the eukaryotic. So there is a blocking of that is a key role in the mitosis. MPF kinase activity requires cyclin B. Cyclin B requires for the chromosome condensation. Here the key word is the condensation, chromosome condensation. After this condensation, the nuclear envelope that is into the vesicles, envelope into the vesicles. Then another case in the cyclin B, its degradation following by chromosome decondensation. That is this cyclin B degradation where it has happened. It's happened at the chromosome decondensation. Cyclin B requires for condensation. If there is decondensation happen in the chromosome, that will degrade. This is the key. This requires ubiquitous to cyclin destruction works in the cyclin B. This process, the envelope formation and exit from mitosis, that process requires the cell destruction box motif in the cyclin B. This RNA is treated as anaphylaxis X is like egg and sperm chromatin were mixed, and MPF activity increases with the chromosome condensation and nuclear envelope breakdown. However, this is not followed by chromosome decondensation and nuclear envelope reform. Here they mentioned they are not followed by chromosome decondensation and nuclear envelope reform because that cyclin D lacking the cyclin destruction box had envelope had been overexpressed. That is the reason here. The next question is very easiest and direct question. Bacterial flagellar motor is the multi-protein complex. This is the complex which is present in the cellular surface of bacteria. Rotation of the flagellum requires movement of protons across the membrane, facilitated by the multi-protein complex that imagine the cellular biology. Then the flagellar motor protein combine to create a proton channel that derive mechanical rotation. In a screen for a mutation, some non-motile ones were selected. Here read the story. First, there is a motor is there in a cell surface of bacteria, that is multi-protein complex. That multi-protein complex needs proton for their activation. The flagellar motor protein is combined to create a proton channel to derive the mechanical rotation. This is the story. In a screen for mutant, if there is a mutant, some non-motile one were selected. This could be how they are selected. The mutation in the charge residue lining the rigid of flagell subunit, flagella subunits. That's a complex protein, right? That complex protein having FLIG subunit. So this is the reason and that question. Okay, next question is cystic fibrosis transmembrane conductance regulator. Protein is known as the cyclic AMP dependent Cl- channel. This is the statement. And CF proteins, cystic fibrosis patients with a mutation of this protein, CFTR protein, shown reduced Cl- reduced permeability, Cl- permeability. Results exhibit elevated Cl- level sweat. In a sweat that having the removal of Cl is higher in the sweat. To prove this, CFTR protein are inserted in a model membrane and Cl transport is followed by radioactive Cl-. It know the topography of CFTR membrane is very important for the function. Despite no proteolytic degradation and denaturation of CFTR protein, wild-type CFTR failed to transport Cl in the liposome. This is the story. Cystic fibrosis is important. Cystic fibrosis transmembrane conductance regulator, that is CFTR protein, is important for the this process. This CFTR protein is known to be cyclic AMP dependent Cl- channel Here the CF patients are cystic fibrosis patients are shown reduced Cl minus permeability, and they are showing a higher concentration of Cl minus in the sweat. To prove this, they are doing the model. They insert in a model that is a liposomal membrane model, and Cl minus transport followed by radioactive minus. This is the procedure. First one is statement, second one is statement, procedure. In this, it is well known. Topology of CFTR membrane is very important and its function. Here, their result, despite of proteolytic degradation, no proteolytic degradation or denaturation of CFTR protein, wild type CFTR failed to transport Cl minus in liposome. This is the result. Here, what they ask, which of the following is correct explanation for this statement, the final result. It is known that topology of CFTR in a membrane is very important for the function. Despite of no proteolytic degradation or denaturation of CFTR protein, wild type CFTR failed to transport Cl minus in liposome. Why it's happened? CFTR protein gets wrongly inserted in the liposome. CFTR protein are wrongly inserted in the liposome. So that is there is no effect. No effect is observed in this model. Okay, this is about that question in the part C unit 2 questions. Tomorrow morning we will start an earlier. I think nearly 8 o'clock I will start. I will share the link today by evening. I will send the link by night. I will send the link. Please join in the remaining 8 classes. Sorry for the delay and sorry for this class extension. Thank you for your cooperation. If you have any queries, you can ask in the chat box and Telegram group. I welcome you to day 23 tomorrow class.